go through this, and I'll start off by outlining what the key concepts are. So that is what I'll be doing, and I just want you to, again, jot down the key big ideas, and just try your very best to sort of follow along and ask lots and lots of questions, please. Okay, so the first dot point that we're looking at, like always, well, not always, because the last module had 3 dot points. This one has 4. So the first one is, what are the causes of infectious diseases? So we're looking specifically at the causes of infectious disease. And our inquiry question is, how are diseases transmitted? So how do they, go about infecting, how do they go about infecting one person to the other? So how does that happen? Now, causes of infectious diseases, basically we're looking at what are these things trying to kill us every second, because there's bacteria and viruses and pathogens everywhere. So we will look at the causes of different diseases, so what the types of murderous pathogens are and how do they go about trying to achieve their target. We will look at transmission. We will discuss an experiment that was done in order to sort of identify how a disease is transmitted and sort of conclude, like what causes diseases, and finally what the impact is on society. So, what is a pathogen? A pathogen may be a word that you have probably heard, or not, probably heard now after the pandemic, but or may not have heard. So a pathogen is any agent that causes or is capable of causing disease to the host. So anything that causes a disease and obviously endangers the host. So the criteria for a pathogen to sort of succeed is that firstly it needs a host in order to hijack the host's cells, or in whatever way they operate. Like a virus does that by hijacking the cells of a host, while something like a macroparasite finds habitat inside an individual and uses their nutrients and whatnot. So it differs based on every pathogen, but generally they need a host, and they can only cause infectious diseases that transmit from one person to the other. They cannot cause non-infectious diseases. So the terms that I'd like you to remember here are microbes. Microbes refer to microorganism. Anything that's small cannot be seen with the naked eye. All pathogens are microbes, but all microbes are not pathogens. We have lots of good microbes for example, in our gut, right? And we need them as well. So not all of them are pathogens. For example, microflora in our gut, not pathogens, play a huge role in helping us maintain good gut health. So let's have a look at the types of pathogens. You'll have to remember what they are and also examples of diseases that they cause. The way they are structured is through like incremental in terms of their sizes. So we start off with like prions, which are like 235 amino acids long. And we go all the way up to macroparasites that can be up to like 25 centimeters long. So it's sort of that progression. So prions are proteinaceous infectious particles, and they are abnormally folded protein, transmits misfolded protein state to other cellular proteins, and an example of this is the mad cow disease. We then have virus, so they're non-cellular, and they're basically nucleic acid. Then you've got the nucleic acid in a protein envelope, and they replicate inside living cells. So what they do is they bind to the host cells, they gain entry into the host cells, and they've got mechanisms that help them do that. And once they're inside the host cells, they hijack processes, and in the case of COVID-19, for example, once it gains entry into the host cells through the use of enzymes in its enzymes on its surface, it gains entry into the host cell and hijacks protein synthesis in order to then control the cell. And if it does that, and and I don't want to go into too much detail, but basically if it that has caused ARDS, that can cause ARDS, which is one of the leading causes of death from COVID-19. So that's like a respiratory disease. So that's how it works, and generally all viruses do that, but obviously differing based on the virus, it differs. So with SARS-CoV-19, the SARS is the virus that causes COVID-19. SARS also caused the first coronavirus that happened, I think, in 2006 2006. And there are other also diseases that it has caused, like the MERS virus in the Middle East. Okay, so an example of that is measles, right? And of course, COVID-19. Bacteria. So bacteria is single-celled prokaryotic organism. It reproduces by binary fission. So hopefully we all know what binary fission is from Module 5. And it secretes toxins, invades cells, and forms biofilms. An example of this is Salmonella. Then we've got protozoa. So now we go into eukaryotic organisms. So single-celled, it's a eukaryotic organisms, and it basically absorbs nutrients from hosts. It secretes toxins, invades cells, and form biofilms as well. An example of a protozoa is malaria, and we will talk about malaria in like 1 second. Finally, we've got 2 more to go. So we've got fungi, which is eukaryotic. It can be multicellular, and it reproduces by spreading spores. An example of a disease, thrush. And finally, we've got macroparasites, which are visible to the naked eye, and they can include ectoparasites, those that live on the organism, and also endoparasites that live within the organism. So things like ticks and tapeworms are an example of that. Okay, so malaria. You'll have to investigate, and this is in the syllabus. So you'll have to investigate the transmission of a disease during an epidemic. So there are a couple to choose from, including like Ebola is another one, but you can sort of choose any, but I've chosen to go through malaria today because it's a really popular one that's really interesting to, I guess, research as well. So malaria is caused by a protozoa, and specifically it is caused by the Plasmodium protozoa. The Plasmodium protozoa is found in the gut of the Anopheles female mosquitoes, and it transmits via vector transmission. So it transmits when an infected female Anopheles mosquito goes ahead and bites a human, and from there, so once they've bitten a human, you've got a mosquito bite, the protozoa the pathogen is transferred into your body. It goes through to your lungs, and the next time another mosquito bites you, let's say another female mosquito that does not have the protozoa bites you, they can take away that protozoa. Like it circulates to them, and that is how the disease spreads. So we've got that was like a little summary. So let's have a look at what actually happens. So firstly, we've got the Plasmodium sex cells, which reproduce in the Anopheles mosquito's stomach, forming zygotes in the cysts of the stomach wall. Then these cysts burst and sporocytes travel to the salivary glands of the mosquito, and from there it is transferred to humans when they bite humans. Sporocytes then travel to the liver, not the lungs, the liver, and they enter the red blood cells and continually multiply. And they can actually cause the red blood cells to rupture as well, because they're continually multiplying within the red blood cells. And infected cells burst, which causes malarial fever. And finally, the cycle continues when the human host is bitten by a mosquito, which passes those mature Plasmodium cells back to the mosquito. Because what happens is, once you have been infected with malaria, the Plasmodium protozoa, it stays within your body, so you can't get rid of it, which is why people see like relapses of malaria, because it stays, it's hard to get rid of, but there have been treatments for it, but now they Plasmodium protozoa has become quite what's the word? It has actually become quite resistant. It has become quite resistant to antibiotics. No, it has become quite resistant to the treatment, and so as a result, that treatment is I mean, people still use it, but it actually is not as effective as when it was first delivered. So you can see in the diagram over there, we've got let me get a pen. I really love using a pen. Hopefully you can see my screen okay. So we've got, okay, here we go. So we've got the transmission to the host that happens here, and then those sporozoites, those are sporozoites, they go straight to the hepatocytes, to our red blood cells, and they affect our red blood cells in the liver. From there, they do mitotic replication. So they are replicating ridiculously fast in the red blood cells, and as a result, our liver cells rupture, and those myriazytes, which are now mature, they are released. Once they're released, what happens is they undergo sexual cycle to produce gametes instead, and you can see here, so they're producing gametes within the human body. So you've got the ring, the trophocyte, schizocytes producing the gametes over here, and then it ruptures again, lets it out, and then from there, once a mosquito bites that human, it goes into the mosquitoes, and the gametes made, they undergo meiosis, they migrate through the midgut wall, forms oocytes, and oocysts, and the sporozoites develop, and the cycle starts again. So with all of this, you can clearly see the way in which it continues to sort of circulate, and it is hard to get rid of. It is a disease that it's hard to eradicate, because firstly, we've got vector transmission by mosquitoes, and then even after you've got the disease, it remains in your body, and that is why people get relapses, malarial fevers, because it gets not reactivated, but essentially, yes, it comes back up, and as a result, the cells start multiplying again, and then they bust and they cause malarial fever and so on. And we will talk a bit about what happens when we look at prevention and stuff later on in the dot point. But you don't need to know in that much detail. You kind of just want to know what I've got here in the dot points for us. All the sort of the terms here, that's just me going into a bit extra detail, because I personally find it very interesting, and I think it's interesting to sort of understand what's happening at the root levels, but you just have to know what the main points are and how it's transmitted. Just because you can get questions which are asking you, how does a named disease transmit and affect people? Obviously it's going to be worded different, but something like that, and that's when you want to be able to name a disease and go through the steps of how it infects everyone. So now we look at an experiment that you will do in class, which aims to investigate the presence of microbes in different water sources. So basically this is just to sort of give you a summary of what it might look like. Obviously the results will differ based on the areas that you test and your sort of experimental design, but basically when it comes to experiments, you want to have clear and concise aims and hypotheses, right? So aim, what are we investigating? Then hypothesis, what will happen? What will we see? Then your descriptive methods you can write out in an exam. So appropriate detail, they can ask you to write out an experiment in the exam. I'm pretty sure they did that in 2021, if I'm not wrong. So something very similar to this. You then want to look at what your expected results are. Obviously, after you've done your experiment, you would know what your results and you want to remember them. You obviously also want to talk about validity and reliability. So Validity and reliability. Validity is the experiment testing the aim, and reliability is, can the results be seen as consistent. As consistent. Accuracy, to what degree are the results precise, and you need to be able to apply these principles to the experiment. Finally, the variables which you need to understand. So having a good understanding of the variables is important. You should be able to tell them apart. Like, especially, I think one thing that always confuses everyone is the difference between controlled variables and having a control. So an independent variable is the one that is being manipulated, while the dependent variable is the one that is being tested. So in this case, our independent variable is the water source that we are using. Our dependent variable is the microbial growth that we want to see in the type of water, so the amount and also the species. We're then looking at the controlled variables, so things that we're keeping the same. So in each sample, we have, for example, the same amount of water, the same temperature at which we are culturing those agar plates, and also doing it for the same time. Finally, controls. We can have a control can be a sealed, unopened agar plate which has nothing growing on it. That can be a control, because ultimately that will tell you that there's no bacteria grown on it because it's been unopened, it's sealed, in comparison to the agar plates that have got the sterilized water, pond water and seawater. So this might differ based on what your school decides to use. Like, if they just want to use, like, I don't know, water from a nearby lake instead of a sea, whatever works. But you'll probably have these 4 sets you'll have these 4 sets essentially. Okay, now we have a look at the modes of transmission. So you've already had, so you've talked about pathogens. We have sort of been introduced to vector transmission when we've looked at malaria. We'll now have a look at the different modes of transmission, and there are essentially 2 main modes of transmission. There is a horizontal mode of transmission and a vertical mode of transmission. Horizontal is when it's direct control. I just like to think of it like horizontal as in proper horizontal, and just helps me think of like direct contact. It can also include indirect contact. It includes vector transmission and contamination. So these 4 are part of the horizontal mode of transmission. Then we've got the vertical mode of transmission, and that includes transplacental transmission, vaginal birth, and also breastfeeding. So for example, with breastfeeding, if the mother has any pathogens, like any pathogens, they can be transmitted to the child. Same thing with vaginal birth and also transplacental transmission. Okay, so now we look at 2 big experiments that really shaped the way in which that really changed our understanding of infectious diseases. Obviously, we now live in a time where our understanding of infectious diseases and the technology that we are using to counter them has increased and evolved significantly in a really nice way. It wasn't the same as when the initial scientists who sort of set out on this path were doing their research. So the first scientist that we're looking at is Louis Pasteur, and Louis Pasteur is he is important because he disproved the theory of spontaneous generation. So people used to think that bacteria or pathogens just spontaneously infect things. He instead said that it's not spontaneous, it's not just happening randomly out of nowhere, there's a reason for it. And he proposed the germ theory of disease, which says that all things come from pre-existing things. And to us now, that is like the most logical thing ever, right? But at the time, because they didn't have the scientific research to back that, they were just like, oh, it just happened. It can just happen. But he said, no, it is happening because it's coming from something that's pre-existing and that has contact with whatever it is that is being infected. And he shows that through his swan-necked flask experiment. So what he did is he took flasks with bent swan necks, and what happened was, with the swan-necked glass, the particles in the air cannot travel through these necks without getting stuck. So if I've got something like, so firstly, let's start here. So I've got two different groups. I start off by applying heat to both groups. So by applying heat, initially what happens is I'm almost, I'm sterilizing it, I'm killing any bacteria in it by applying heat to it. I then let the flask sit for a while, but with one I let it sit while it's closed, so while it is closed, but with this one I open the flask, so I'm removing the neck and I'm letting it sit. When that happens, that means that suddenly all the microbes in the air have access to this solution right here. So as a result, all the microbes from the air are going to come in, and that is going to lead to bacteria being present in the flask. However, if I let the flask sit and I'm not removing it, I'm leaving it as it is, because of this passageway into the flask, you would see that there's no bacteria present, because bacteria cannot, come in through here, travel upwards and go down here. It gets stuck here on the side. So as a result, there's no bacteria present. And yeah, so basically apply the heat, break the neck, observe the growth. Flask with broken neck means that there's microbe growth, and flask with without the broken neck means that there's no growth. And that basically tells us that the it's not spontaneous generation. It's basically that the disease and the bacteria is coming from pre-existing microbes in the air that get access to these solutions right there. Okay, next we look at Koch's postulates. So this is where he developed a procedure for isolating and identifying microbes which cause disease. So he wanted to sort of see what is whether or not it is microbes that are causing disease. So what he did was, in all organisms with the disease, he says that, let's logically work through his steps. He said that in all animals that have a certain disease, there are going to be microorganisms, microbes present that are going to cause that disease. Those microbes can then be isolated and they can be grown in pure culture. So if I've got postulate 1 where the same microorganism, so I'm trying to prove the same microorganisms are present in every case of the disease. So if the symptoms are the same, more than likely there are going to be same microorganisms that are infecting these organisms. So here my potential microorganism is the Anthrax bacilli. So what happens is I'm going to isolate that microorganism and grow it in a culture. So how do I isolate it? I isolate it from a dead from a tissue of a dead animal, and I make a pure culture. So I grow it in like an agar plate in my culture. Then what I do is I take the culture and I take the microorganisms from the pure culture that I've grown, and I inoculate that into a healthy, susceptible animal, into another animal. Not the most ethical thing to do, but hey, that's what he did. So when we inoculate a healthy organism with a pure culture, it develops the same symptoms as the original organism. Because it's got those same symptoms, right? What that tells us is that ultimately it's that same microbe is causing that disease. However, what we do is we isolate and regrow that microorganism from that newly infected organism, and we do the process one more time. And so if same, then we realize that, hey, it's that microorganism that is causing the disease. So like I said, not the most ethical experiment, but that is how he was able to prove that microbes are causing disease. So every time you see similar symptoms, more than likely it's the same microbe.